video, we're going to be looking at the grade 12 genetics section. First section we're going to look at in genetics is concepts in inheritance. And the key terms for this section are as follows: chromatin and chromosomes, genes and alleles, dominant and recessive alleles, phenotype and genotype, homozygous and heterozygous. You need to be able to identify the differences as well as the similarities between each of these terms, and you need to be able to apply them in context. So let's start with chromatin versus chromosomes. When we look at a cell, we can see at different stages on a micrograph, the nucleus looks like that, or the nucleus looks like that. In this nucleus, it's very difficult to see the DNA, and that's because the DNA is unwound. But in this nucleus, it's possible to see individual strands. And therefore, this DNA here, we call the chromatin or chromatin network, and this DNA over here, we call chromosomes. It's identical stuff, it's made up of exactly the same stuff, but the way in which it appears is different. So what is that difference? Well, when we look at chromatin, the chromatin is unwound. And we only really find chromatin during interphase. When that chromatin condenses, though, what happens is that the DNA, here's our double helix, is wrapped around these proteins that are called histones, and that in turn is coiled up on itself, making those little balls. And then it's coiled even further so that these little balls eventually become very tightly wound. If you have a landline in your house and you've got that curly telephone wire, you will know how that curly telephone wire can sometimes get curled up on itself. It's exactly the same setup here. And these coils then get further and further coiled until eventually we end up with a highly coiled structure, which is called a chromosome. The chromosomes are only visible during cell division, in other words, during mitosis and meiosis. So there are three words that are fairly similar, and unfortunately in the exam, where you have biological terms that are similar, you have to be able to spell them correctly and use them correctly. That's one of the exceptions to the rule that spelling doesn't count. So you must know the spellings of these three words and not get them confused. So, chromatin is the material from which the chromosomes of eukaryotes are composed. And it's made up of proteins, RNA, and DNA. Then you've got a chromosome, which is a thread-like structure, which is made up of the nucleic acids and proteins that are found in the nucleus. And the thing that we focus on about chromosomes is that they carry the genetic information, the inheritable genetic information in the form of genes. We're also going to learn about chromatids, or you will have done under the DNA section that we did. Chromatids are the two thread-like strands. So when you look at your chromosome, this is what we call a double-stranded chromosome. It's made up of two chromatids. There's one chromatid, and there's the other chromatid. And so those chromatids make up the chromosome. Okay, what about genes and alleles? So a gene is a portion of a chromosome which codes for a specific characteristic or part of a specific characteristic. In actual fact, there are very few characteristics in our bodies that are coded for by a single gene. Most characteristics in our body are what we call polygenic traits. There are many genes which together code for that characteristic. What's important here is that this characteristic must be heritable. In other words, you can inherit it from your parents, and it is located in a specific place on a specific chromosome. It's not random. Now an allele, not allele, allele, an allele is an alternative form of that gene. For example, blue eyes versus green eyes versus brown eyes. Those are alleles of the same gene. Those genes are found in the same place on homologous chromosomes. So pair 1, for example, or pair 12. the genes that occur on the first chromosome in pair 12 will be the same genes that occur on the other one, but the alleles will be different. The versions of that gene will be different. So if we look at a chromosome like this and we unwind it, a portion of that will be a gene, but what version of that gene you have will be different. So for example, you could have a gene that gives you freckles and a gene that doesn't give you freckles. They're on the same chromosome pair and they're at the same location. And therefore, these are alleles of the same gene. Now, just for interest's sake, some of you may be aware about the Human Genome Project. What the Human Genome Project does, or has done, is that it has mapped all the genes that occur in the human DNA. And they've created these posters of each chromosome along with the various disease genes that can exist on that chromosome. So this is, for example, chromosome number 7. And you can see here, there's the gene for osteoporosis, growth hormone deficient dwarfism, polydactyly, that's having more than 5 fingers or 5 toes, the gene for colon cancer. Here's another gene that contributes to osteoporosis, cystic fibrosis, obesity, having faulty taste receptors. So these are just the disease genes that you might have, not to say that you will have, but that you might have on chromosome pair 7. And if you're interested in more, you can go and have a look at this website here. If you search for the word gene specifically, you'll be able to have a look at all the posters and have a look at where the different disease genes are found. Now, when we talk about alleles, that brings us back to Mendel and to talking about the law of dominance. The law of dominance states that if you have both a dominant and a recessive allele present, the dominant is the one that will show up. What on earth does that mean? A dominant allele is one that is expressed. So you have 2 versions of the same gene, freckles and no freckles. The one that shows up, in other words, whether or not you have freckles, will be the dominant. The one that only shows up if there's no other alternative is the recessive. So we say that dominant genes are always expressed if present, and when we write them down, we write it with a capital letter. The recessive allele is the one that is only expressed if there is no dominant gene present, and therefore we always write it with a small letter. So, genotype. This is what is in your genes. This is what specific genes do you have present on that chromosome. It's the complete set of genes an individual has, or it's the alleles for a specific trait, freckles and no freckles. That genotype codes for a particular phenotype. Notice P for phenotype, because phenotype is your physical appearance. So if you have freckles, your phenotype, you must have a gene for freckles, and that is your genotype. If you have no freckles, your phenotype, the gene that you have will be the recessive gene, the no freckle gene. So how do we write this? Well, if you have the dominant gene on both of your chromatids, or both chromosomes in the pair, then you will write them with a capital letter. If you have 2 recessives, you'll write them with 2 small letters. And if you have different versions, different alleles, then you write 1 capital and 1 small. So let's say that capital B represents having freckles. And the gene that you have there, so that's your freckle gene, the allele of that at this point is the dominant one, capital B. And the allele on the other one is also the dominant one, capital B. Here's the freckle gene, but which version do you have? You have the no freckle version. Same thing here. What gene do you have? You've got the gene for freckle, but the version of that gene that you've got is the no freckle version. Or alternatively, If you've got one version of that gene that gives you freckles, one version of that gene that does not give you freckles, then you have a capital B and a small b. Now it becomes a bit of a mouthful to say you've got capital B and capital B, or capital B and small b. And so we've come up with words to express this. Homozygous, homo meaning the same, means that the letters that you have are the same. In other words, both alleles on your chromosomes are the same. Heterozygous, hetero meaning different, means that the two versions that you have, the two alleles that you have, are different to each other. And you can just say heterozygous, and everybody who knows that term understands that it means you have one capital and one small. With homozygous, it's a little more tricky because you need to indicate to your hearer whether it's homozygous dominant, in other words, you have the dominant version for both your alleles, or whether it's homozygous recessive, two small letters for both alleles. And so we write it like this. If we look at purple flowers, that's the phenotype, what it looks like, the physical appearance. There it's capital P, this is homozygous dominant. If you have white flowers, white being the recessive allele, it means that you are homozygous recessive and your genotype is small p, small p. Remember, the recessive allele is the one that only will appear, only shows up in your physical appearance if the dominant gene is not present. And in this case, purple is the dominant and white is the recessive. If you have a heterozygous condition, one capital, one small, so you've got one gene that says purple flower and the other allele that says white flower. What happens now? This is where the law of dominance comes into play. Because we have one dominant gene, if the dominant gene is present, the law of dominance says, it will be the one that is expressed. And that's what we see here in the phenotype. So in the genotype, I've got a heterozygous condition, and the law of dominance says that therefore the dominant gene is going to be expressed. And so in the phenotype, the physical appearance of this flower, it will be purple. Let's move on and talk about the monohybrid cross. Earlier I mentioned Mendel. Who was Gregor Mendel? You can see a photograph of him here. He's quite clearly a monk, but he was a monk who was interested in science, and he did a whole lot of experiments using pea plants. He's known as the father of genetics because of what he discovered. His powers of observation were phenomenal. He was very patient, and he did thousands and thousands of experiments that were very, very carefully controlled. In his experiments, he looked at 7 different traits of the pea plant. You don't actually have to know all the different traits, but it's helpful if you can remember many of them. He was very fortunate when he was studying them that all of these traits are coded for by just one gene. If you remember earlier I said most of our characteristics are actually polygenic traits. They're coded for by more than one gene. But all of these 7 are actually coded for by just one gene each. And he was very fortunate that he chose that because having these particular characteristics, when he did his experiments, it really helped him to get to the bottom of how inheritance of genes and genetics occurs. So you can see that he listed the dominant and the recessive traits. So purple flower color is dominant, meaning if you have one gene, one allele that is a capital P, it will show up and your offspring will be purple. And the recessive of that is white. When looking at your plant height, tall was dominant over short. He then looked at seed color and seed shape. Yellow is dominant over green, and round is dominant over wrinkled. When he looked at the pods, green pods were dominant over yellow, and full pods or inflated pods were dominant over the constricted. And lastly, he looked at where the flowers were present on the plant. If they were at the top of the plant, axial, that was dominant over terminal, which was on the side of the plant. Now, when he did his experiments, he obviously needed a shorthand to be able to record what happened. And that is known as the monohybrid cross. Mono, one, hybrid, trait. So he was looking at one trait at a time, and he developed this monohybrid cross to represent his experiments. In the exam, you are expected to follow a particular template to fill out or to complete a monohybrid cross. If you don't use the template, you will lose marks. When looking at monohybrid crosses, it's important that you understand the law of segregation. Now, the law of segregation says that when gametes are produced, the two copies of each hereditary factor will segregate so that the offspring acquire one factor from each parent. That was the terminology that Mendel used. Nowadays, because we understand what's going on in meiosis and because we have a better understanding of genetics, we would say that during the production of gametes, the alleles, which is each hereditary factor, or the chromosomes carrying each allele, segregate. That means that they move apart from one another. They separate themselves from one another. And that's exactly what happens in meiosis. So in metaphase 1, the homologous pairs line up at the equator and then they move apart from one another. And the result is that the offspring acquire one allele from each parent. That's because the gametes are haploid. And each of those gametes that's haploid, when it then fuses together to form a zygote, means that the zygote has two copies of every allele, one from each parent. If you haven't done meiosis yet, this will make a lot more sense once you have. So, homologous chromosomes separate the end of metaphase 1 into anaphase 1, which means that the alleles for each are separated. And therefore the gametes contain only one chromosome from each pair, and therefore only one copy of each gene, one allele of each gene. Now the Punnett square is part of the monohybrid cross, and you can use that, for example, in section A, where you've got your multiple choice questions. You can just draw a little Punnett square on your exam paper or on the question paper to quickly work out what the answer is going to be for the monohybrid cross. But in section B, you will need to do the entire template of a monohybrid cross. You cannot just do the Punnett square. You will lose marks if you just do the Punnett square bit. So the Punnett square is used to determine what happens at the fertilization of the gametes. And the way we do this is we put the alleles on from one parent on top and the alleles from the other parent on the left. So let me show you how the Punnett square works first, and then we'll go and have a look at the full monohybrid cross template. So here we have the male parent. There you can see a particular homologous pair of chromosomes. That is the position of the genes that we're looking at. And you can see that this male parent has two different alleles. He is heterozygous, one capital, one small. Here's the female. There is her chromosome pair, the homologous pair. There are the genes that we're looking at. And again, you can see that she is also heterozygous, capital and small. So we indicate, we write the capital B there, the small b there. And the reason we separate them out is because in the sperm, these two chromosomes will separate from one another during anaphase 1, and therefore the gamete will only contain one of each, and therefore only one letter of each of those two alleles. Likewise for the female. So one of the gametes, one of her eggs, will have the capital, and the other will have the small. Now we need to look at what happens during fertilization. If this sperm cell fertilizes that egg cell, the offspring will get a capital B from the father and a capital B from the mother. There they are. If that sperm cell fertilizes that egg cell, then we've got a capital B from the father and a small b from the mother. And that's what we can see there. Same procedure here. There's the capital B from the mother. There's the small b from the father. And in this last square here, there's the small b from the mother and the small b from the father. So we have a homozygous dominant, a homozygous recessive, and two heterozygous. Now notice the fractions that are here. 1 out of 4 options is your homozygous dominant. 1 out of 4 options is your homozygous recessive. And 1 plus 1, 2 out of 4, in other words, 50% of your options will be heterozygous. Okay, let's look at these three examples of a Punnett square. So if we take this first one, we've got a dominant and a recessive. In other words, the parent here would have been heterozygous. Dominant, recessive, the parent here would have been heterozygous. In this one, we've got a heterozygous parent and a homozygous recessive parent. In this one, we've got a dominant homozygous, and in this one, we've got a recessive homozygous. So let's go through them step by step. So what are the ratios? Well, remember we have a convention that you always write the capital letter first. So that one would be capital T, capital T. That is what that offspring would be. If this sperm cell joins with that egg cell, this is what it would be, a heterozygous offspring. If this egg cell and that sperm cell fuse together, this is the offspring, heterozygous. And again, remember the convention, we always write the capital letter first. And the last option, small t, a homozygous recessive individual. You will be asked to write this out as a genotype and possibly or probably as a phenotype. So if we look at the genotype, 1 in 4, which is 25%, is capital T, or homozygous dominant. 50% are your heterozygous, capital T, small t, because 2 out of the 4 squares are your heterozygous condition. And 25% is small t, because you've only got 1 out of the 4 that is the homozygous recessive. You can also write this as a ratio. In other words, 1 to 2 to 1. 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. 1 to 2 to 1. Pause the video now and see if you can do this one on your own, and then start playing again to see the memo. Did you remember to write the capital letter first? Did you get it all right? Let's try another one. Pause the video and play when you're ready to do it. In this example, all 4 of the offspring are heterozygous. When it comes to writing out your genotypes, we would say that 100% are capital G, small g. So let's talk now about the template that you have to use in the exam. In the exam, you are expected to list all of these things down the left and to fill them in correctly to get the full set of marks. And a question like this could be 6 to 8 marks. You start with the parents, and the parents we call the P1 generation. And then you move on after fertilization to talk about the offspring, known as the F1 or F2 generation, depending on how many times you have to repeat this Punnett square. F stands for filial, which means offspring. Let's look at an example. Notice that marks are given for this template for putting in the words meiosis and fertilization. If you put those 2 words in, you will get a mark. In the correct place, that is. Likewise, just for indicating P1 generation and the F1 generation, assuming you've got them in the right place, you will get a mark. You haven't filled in any information yet on the template, but because you've written the template down and you've written it down correctly with all the words in the correct places, you've already got 2 marks. All right, so let's fill this in. Let's say we are looking at fur color. The characteristic we fill in, fur color. And you should be given the key in the exam question. If not, you can use whatever letter you like. I wouldn't recommend that you use S's or L's because they're very difficult to tell the capital and a small letter apart. So let's say we're going to have black fur, which you're told is the capital because it's the dominant allele. And that's going to be, let's say, versus white fur, which is the recessive. Now, because we are looking at a single characteristic, you must use the same letter, just a small letter. So the key here would be B, small b. In the question, they will tell you whether the male is black or brown or white or whatever it might be. They will give you information that you then have to interpret to be able to work out what the phenotype is, or to be able to work out what the genotype is. So there is some interpretation required in these questions. So let's say we have a black male, and again, they should tell you in the easier questions, they should tell you whether it's heterozygous or whether it's homozygous dominant. And let's say that that is crossed with a white female. Now, if you've been told in the question that the genotype is homozygous dominant, it would obviously be capital B, capital B. White, because we know that white is the recessive color, they don't have to tell us what the genotype is because we know the phenotype. If the phenotype is the recessive, remember the law of dominance, only shows up when there is no dominant allele present. So white must be little b, homozygous recessive. Meiosis takes place. Chromosomes segregate from one another, which means that the alleles are independently assorted. And so as a result, each gamete from the male will contain one capital B, and each gamete from the female will contain one small b. Now we do our Punnett square. It doesn't matter which way around you put the male and female. What's important is that you keep the two gametes together. So one parent on the top, one parent down the side. And then we fill it in. And then the last step is that you must fill in the phenotype and the genotype. Genotype is easier to start with because you've got it sitting in front of you in the Punnett square. And you can see here that all four of these are capital B, small b. So my genotype is going to be 100% capital B, small b. Now I have to interpret that as a phenotype. If this is a capital B here, the law of dominance says that if a dominant gene is present, it will be expressed. So in the phenotype, I'm not going to see the recessive, I'm only going to see the dominant. And the dominant, if we go back to our key over here, the dominant is the black color, which means that all of these individuals here are going to be 100% black. Occasionally in exam papers, instead of doing the monohybrid cross in a forwards direction, so talk about the parents, and then you do the offspring, and then you write the offspring's genotype and phenotype, they want you to work backwards. So they will say to you, they'll give you the genotype of one of the parents, and the other parent, they will only tell you the phenotype, and you will need to work it out. The way that you work it out is by doing multiple monohybrid crosses and then coming to a conclusion. Remember when you do that, you must actually write your conclusion down. So here we have an example. We've got two possible outcomes. We've been told that we have, let's say, for example, a tall, T for tall, a tall parent and a short parent. Now the short parent, because that's recessive, we already know what the genotype is going to be, small t, because it's homozygous recessive. That's the only time you will see the recessive characteristics show up in the phenotype. But for the tall parent, we don't know. Are they dominant, homozygous dominant, or are they heterozygous? Because both being heterozygous and being homozygous dominant will make the dominant gene be expressed in the physical appearance. As long as you have one capital letter, that is the characteristic you're going to see. And so from the phenotype, you can't tell what the genotype is going to be. So I'm going to have to do two Punnett squares, two whole monohybrid crosses, and then write a concluding statement. All right, so I've done my 2 possible monohybrid crosses. Now I go back to the question, and the way the question is phrased will tell you what they're looking for. So, for example, if they said to you that you've got a tall individual crossed with a short individual and all of the offspring were tall, looking at these 2 Punnett squares, which is the correct parent pairing? Which is the correct genotype for the 2 parents? Well, obviously, if you've got a tall and a short individual and all the offspring are tall, it means that the parents must be homozygous, both of them. One will be homozygous dominant, one is homozygous recessive. So at the end, I would then say the correct parent genotypes to achieve this must be big T crossed with small t, for the parents. If, however, they said to you they have a tall individual and a short individual, and only 50% of the offspring were tall, what's the genotype of the parent? Well, here's 50% of my offspring being tall, and therefore my parent must be heterozygous. So in a test cross, you have to do multiple monohybrid crosses and work backwards to work out what the possible genotypes of the parents are. It's good to remember that if all the offspring express the dominant allele, then your unknown individual, the parent that we're trying to find, must be homozygous dominant. However, if there's a split and some of the offspring show the dominant characteristic and others of the offspring are the recessive, then the parent that you're looking for must be heterozygous. Knowing that, though, is not enough. You have to be able to show it by actually doing 2 complete monohybrid crosses and writing a concluding statement at the end based on whatever the question is asking you.
So the genotype of a plant that results from a cross between a plant with red flowers, capital R, and white flowers, small r, will be. Let's do the Punnett square. So if we draw our little Punnett square, we're going to put the gametes from one parent at the top and the gametes from the other parent on the side. Remember that you split the chromosomes, they segregate separately, and so we split the capital R and the capital R. Likewise the small r and the small r. Now we fill in our Punnett square. And so we can see that all of them are capital R, small r. Remember that you need to always write the capital letter first, so it cannot be D, it must be R. Let's look at question 4. Choose the correct cross if the result is 50% homozygous dominant and 50% heterozygous in the F1 generation. The fact that they've mentioned the F1 generation rather than the parents means that this is a test cross and you're going to be working backwards. So let us do our Punnett square. And we're going to fill the Punnett square in first. It says 50% are homozygous dominant. That means 50%, 2 out of 4. Homozygous dominant, capital letter, capital letter. So I can use any code I like. I'm just going to stick with the R's. So 2 of them are capital, capital. 50% are heterozygous. So 2 out of 4 are heterozygous, different alleles. Now I need to fill in what the parents would be. So one of these R's must come from there, and the other R must come from there. Likewise here, 1 from the top and 1 from the side. When I now look at the last column, that capital obviously comes from that side, which means the little r must come from there. Likewise here, use this one to check. That big R comes from the big R, the little r comes from the little r. So I know that my parents now are this. And I look at the answers. Okay, they've used B's for their answers. But I need one that is dominant, homozygous dominant, with one that is heterozygous, which means that it must be B. Let's look at question 5. 2 white heterozygous cats were crossed. Stop there. I've got white heterozygous cats. Heterozygous means different letters, so that. The fact that that genotype gives you a white phenotype means that white is dominant. Okay, let's carry on reading the question. So 2 white heterozygous cats were crossed, where white fur is dominant. There we go. They've told you the information, but you actually didn't need to be told that because you could work it out. The alternative is black. So black is going to be your recessive. Now they want you to work out what the correct phenotype is of the F1 generation. So let's draw our Punnett square. And we've got capital R, small r, crossed with capital R, small r. I fill that in. What can we see? We've got this one, 1 to 2 to 1. So we've got 1 homozygous dominant. We've got 2 heterozygous. And then we've got our 1 homozygous recessive. But that is the genotype. We must interpret that as a phenotype. And in our phenotype, this one and this one and this one all have a capital letter. The law of dominance states that if a capital letter is present, if the dominant gene or the dominant allele is present, it will be expressed. So I've actually got 3 of my 4 options, and 3 out of 4 is 75%. So I've actually got 75% that are going to show me the dominant, which we know in this case is white. So I'm going to have 75% white with 25% black, which means C is my answer. Question 6. A heterozygous red flower plant is crossed with a homozygous white flower plant. Yields 300 plants. Okay, let's stop there and interpret the information they've given us. We've got heterozygous red. So we've got RR, and the phenotype is red, which means that red must be the dominant color. And it's crossed with a homozygous white. Now, if capital R is red, then white must be little r, little r. And it says it gives 300 plants. Okay, so let's do our Punnett square. So I've got capital R, crossed with small r, and I fill that in. So 50% of these, because they carry this dominant gene, 50% are going to be red, while 50% are going to be white. Now they said to you that there are 300 new plants. So 50% of 300 is 150. So 150 of the new plants are going to carry white flowers. One last question. The babies of a purebred white rabbit crossed with a purebred black rabbit were crossed. Okay, now the term purebred in a question like this means homozygous. So here we have a homozygous white with homozygous black. And you're told that black dominates over white, the offspring of the F2 generation. Now when it gets to the F2 generation, you need to obviously do the F1 generation first. So we have a purebred white, which we're told is recessive. And we've got a purebred black, which is obviously dominant. Let's do our Punnett square. So this is F1. You can see that all of them are now heterozygous. If we now take these offspring and we cross them again, so this time we've got 2 heterozygous individuals, This will be our F2 generation, the grandchildren. Okay, so now let's look at the genotype. Genotype here, we've got 1 homozygous dominant, 1 homozygous recessive, and 2 of the heterozygous. 1 to 2 to 1. If we look at the options that we have now for our offspring, we've got all black, all white. Well, it's not any of those. And then we've got 75% black or 75% white. Again, remember you've got 3 out of your 4 options that have at least 1 capital letter, and therefore will show the dominant characteristic. That's the law of dominance. And capital R in this case represents the black hair, because we told that black dominates over white. So we will have 75% black and 25% white, which is answer C.
The last topic we're going to look at now is an extension of the monohybrid cross, the dihybrid cross. Let's say, for example, a tall plant with purple flowers is crossed with a short plant with white flowers. What would their offspring look like? The law of independent assortment in practice, when we consider 2 traits on 2 different chromosomes, dihybrid, how does that work? If we imagined that an individual only has 2 pairs of chromosomes, during metaphase 1, those chromosomes could either align like this, with the maternal chromosomes on one side and the paternal chromosomes on the other, or like this, where they get 1 maternal chromosome and 1 paternal chromosome. Mendel was very fortunate that all 7 of the traits that he chose actually ended up on different chromosomes, which meant that when he wanted to look at a dihybrid cross, it worked. There were no complicating factors. Likewise, as I mentioned earlier, he was very lucky that each trait he looked at only is coded for by 1 gene, which made it easy to work with. If we were to work with all 23 chromosome pairs, scale it up, we would be looking at over 8 million possible combinations in terms of the way that those chromosomes align at metaphase 1. However they align at metaphase 1, that obviously influences what happens in anaphase 1 and eventually after meiosis is complete in terms of the genes that are present in those gametes. So here you can see we have capital A on that one, and the other allele is a small a. So the dominant on one chromosome, the recessive on the other. And they will end up in different chromosomes. Likewise, you've got a dominant and a recessive, which will end up in different chromosomes. In this one, because of the way that these chromosomes have aligned with 1 paternal and 1 maternal chromosome on each side, the dominant ends up here, the recessive there, but here is the recessive and there is the dominant. And so we have a different gene combination there, which means that in order for us to actually understand what's going on, the dihybrid cross, you're always going to go to the F2 generation. With the monohybrid cross, you only need to go to the F2 generation to interpret what's happening if under a very certain sort of set of circumstances. But with the dihybrid cross, you have to always go to the F2 generation before you can start to see any pattern in terms of inheritance. So what you have to remember for the dihybrid cross is that the way we write these things is very similar to the monohybrid cross. Here you can see we've got HHBV. HH being the first characteristic we're looking at, BB the second characteristic, and this individual is homozygous dominant for both traits. Now you can be homozygous dominant for both, or you can be homozygous recessive for both, or you can be homozygous dominant for one and recessive for the other, homozygous recessive for one and dominant for the other. Those are the combinations that your parent generation are going to have, P1. Whichever of those combinations you have, your offspring are going to be heterozygous. If however, they give you these two, then it will be slightly different. And the trend of late when it comes to setting the exam is to do exactly that. So in the past, when we tested dihybrid crosses, we would always give you an easy one like that or like that, where your offspring in the F1 generation were always heterozygous. Of late, the last year or two, the trend has been to give you those, for example, or those, or those, or those, as a combination of parents, because that makes things a little bit more interesting in the F1 generation and then obviously in the F2 generation. With a straightforward example though, because your F1 generation are always heterozygous, when we crossbreed those individuals, your F2 generation, the grandchildren, they have a phenotypic ratio 9:3 to 3:1. Set in stone, that's how it's going to be. So let's have a look at an example. Yellow seeds and green seeds, there we're looking at the color of the seed. Smooth seeds and wrinkled seeds, that's looking at the texture of the seed. So here's our round yellow, notice that's homozygous dominant for both, and our green wrinkled, homozygous recessive for both. Mendel would have called those pure breeding. When you look at an individual like this, for the F1 generation, remember, one of those R's has to go into each gamete, one of those Y's has to go into each gamete. And so the gametes will be capital, 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 capital. Likewise here, one of the R's has to go into each, one of the Y's has to go into each, and so you end up with R, Y, both small or recessive ones. You fill in your Punnett square exactly as before. Capital R, small r, capital Y, small y, capital R, small r, capital Y, small y. And likewise with those. And so you can see that your offspring for F1 generation in this case are 100% heterozygous for both conditions. We crossbreed those individuals. And when we crossbreed, we now have to work out what the gametes are going to be. And so we use FOIL, exactly like you do in maths for factorization. F is for first, outer, inner, last. So we take, don't worry about the P's and G's, don't let the change in code confuse you. There's our one characteristic, there is our other characteristic, heterozygous for both conditions. So we take the first of each, we take the outers, we take the two inner ones, and we take the outer one, the last one of each of our conditions, which gives us that. So these are going to be the 4 possible gametes that we can have. And we put that down the side of the Punnett square and we put it across the top, because when we crossbreed, we are breeding within the same set of offspring. So if you think of plants, the offspring of those plants, we simply take 2 of those offspring and we pollinate them with each other. So we fill in those 4, first, outer, inner, last, first, outer, inner, last, down the top and across, down the side and across the top. And then we fill in the Punnett square exactly as we would for a monohybrid cross. So you've got capital R and capital R, capital Y, capital Y. Down the bottom here, you've got small r, small y, small y. And so there's a homozygous recessive for both, homozygous dominant for both. Now, if we were to mark out all of those that in the phenotype show the dominant characteristics, there are actually 9 of them. This is the only one in the genotype that is homozygous dominant, but in the phenotype, there are several that have at least 1 capital letter for both conditions, for both traits. We look at another example. They've actually color-coded it for you. So this example is looking at cat fur. And here you can see in the F2 generation, in the grandchildren generation, all your purple squares will have the dominant traits in their phenotype. They have at least 1 capital S for the texture of the fur, and at least 1 capital B for the color of the fur. If you look at the green square, that's the homozygous recessive. It's the only one that is completely recessive for both characteristics. Then you've got 3 peach and 3 blue. The 3 peach have the dominant characteristic for the texture, but the recessive characteristic for the color, and the 3 blue are the other way around. They have the recessive characteristic for texture, but the dominant characteristic for color. And so therefore, your ratio here is still 9 to 3 to 3 to 1. And remember that that ratio is a phenotypic ratio. In other words, what you see in the physical, not what you see in the genes. If you find the dihybrid cross very confusing, don't panic. It's only asked in section A, where it's a very small number of marks, 2 or 3 or 4 marks. But you must understand the monohybrid cross and be able to do the monohybrid cross, because that's asked in both section A and in section B. And it is worth a significant number of marks.